On behalf of DNA Genotek and Diversigen, I would like to welcome you to Progress of Omics in Microbiome Research webinar. Some housekeeping before we start. Each presenter will share a case study, followed by Q&A session. During the presentation, feel free to submit your questions using the Q&A function that is at the bottom of the screen. We will compile the questions, and at the end, we will have the presenters answering during the Q&A session. Back to the webinar. The integration of two or more omic data sets has opened the door to generate unprecedented understanding of biological processes. These multi-omic approaches promise to accelerate all aspects of life sciences, from academic discovery to clinical applications, including better biomarkers and more effective intervention, and hopefully one day helping us to live long, healthy lives. Today, we will present multi-omic case studies showcasing microbiome, metabolomic profiles, and many other omics. We will discuss some aspects of study design, project execution, as well as data analysis and integration. For today's discussion, we have three outstanding panelists. Dr. Tonya Ward, Director of Data Science and Bioinformatics at Regression. Tonya has ample experience in microbiome research, creating bioinformatic tools for analysis, leading local clinical research studies, and collaborating on numerous multi-center initiatives. Our second panelist, Dr. Greg Michelotti, Scientific Director of Biology at Metabolomics. Greg has considerable track record in academia and industry. He has generated dozens of relevant peer-reviewed publications and leads Metabolome scientific vision. Finally, Dr. Nima Agepur, Assistant Professor at Stanford University. Nima's laboratory develops machine learning and artificial intelligence methods to study clinical and biological modalities in translational settings. His primary interest is leveraging multi-omic studies and wearable devices to address global health challenges. With this in mind, and no further delay, I will let the experts in the topic start. So, Tonya, you are first.
Awesome, thanks, Carlos. So first, I just want to say thank you to the organizers for having me on this panel today. And today I'm going to be talking about a rather large study that I was fortunate to be a part of. So to preface this, I want to give a shout out to Perna Kashyap and Dan Knights, who are kind of the brain children behind the study, as well as our many collaborators that worked very hard to find the results that we found here. So today we're going to go over a multi-omics approach that we took to explore mechanisms behind irritable bowel syndrome, or IBS. So what is IBS? IBS is recurrent abdominal pain and discomfort. It changes in stool form and frequency. So that could be constipation, diarrhea, or a phenotype of mixed. Symptoms are affected by diet, host genetics, and the environment. And there are periods of remission, which can make it really hard to study in terms of a cross-sectional design because these phenotypes can be changing. So that's why we wanted to integrate multi-omic measurements with longitudinal tracking to try and identify IBS subtype specific mechanisms behind IBS. This is an overview of our study design. So we had patients with IBS-C, which is IBS with constipation, IBS-D, which is those with diarrhea, and healthy controls. We sampled them longitudinally for a period of 7 months. So we collected stool, and that was slated for metagenomic and metabolomic profiling. We had dietary recalls that corresponded to those stool collections. And we also had biopsies at the beginning and the end of the study. Those biopsies were slated for 16S metagenomic profiling, metabolomics, host transcriptomics, methylome and cytokine panels, as well as host physiology through microscopy. We also had serum samples that were taken at the beginning of the study, and those were slated for metabolomics and a cytokine profile. If we first look at the metabolomics data, what we found is that those with IBS-C had significantly decreased levels of lysine, uracil, and hypoxanthine. What's really interesting is the hypoxanthine was actually decreased also in those with IBS-D. So hypoxanthine can serve as an energy source for intestinal epithelial cells, and it promotes intestinal cellular barrier development and recovery following injury. So this lower amount of hypoxanthine could reflect decreased production by the host, so in the intestine, or perhaps elevated breakdown of hypoxanthine by the microbiome in the gut of those with IBS. So the next thing that we looked at in terms of all of our omes for this study was the functional profile that we derived from the shotgun metagenomics that we did on the stool samples. So because it's shotgun sequencing, we're able to get a really accurate representation of the genes within the bacteria in the microbiome. So what we found was that there are two types of xanthine degradation enzymes that were in fact in higher abundance in IBS patients, including xanthine dehydrogenase, which is this KO87 in the KEGG hierarchy. So if we have a look at kind of the overall picture for this hypoxanthine involved pathway, it's actually part of a purine degradation pathway. So purine salvage happens in our bodies, and we know that hypoxanthine is decreased in those with IBS. So that's the middle box there with the arrow pointing down. And we found that via metabolomics. We also know that the xanthine oxidase is higher in relative abundance in the bacteria of those with IBS. So those would be the blue boxes there. Together, this suggests that the microbiome in IBS patients exhibits an increased capacity for hypoxanthine utilization and breakdown. But the next thing that we wanted to explore was the host transcriptomics that we had. So when we look at the intestinal cells themselves, we know that they have a limited capacity for de novo synthesis of purines. And instead, they use that salvage pathway. So when we looked at the host transcriptomics via targeted RNA-seq, we found that we have a decrease in the hypoxanthine. And using the biopsy samples, we found that purine nucleotide phosphorylase, or PNP, is the first gene in that purine salvage pathway, was actually increased in those with IBS. We also found that xanthine dehydrogenase in the host cells, so XDZ, was also increased in the intestinal epithelial cells. What's really interesting is that this is via host transcriptomics, so RNA-seq. We also did a genetic panel to look for SNPs in these individuals, and we didn't find any correlation with the polymorphisms in those with IBS in comparison to healthy controls in any of these genes that are associated with the pathway. So it does seem like it's at the transcript level and not the genomics level that this might be happening. So let's go back to our pathway. So we know that the hypoxanthine is decreased in the stool via metabolomics in IBS and IBSD. We know that the gut microbiome has an increase in the amount of genes for xanthine dehydrogenase. We also know that the host, so intestinal epithelial cells, have an increased amount of PNP being transcribed and also an increased amount of xanthine dehydrogenase being transcribed. So if we put this all together, we actually have a new target potentially for a pathway for treatment of IBS. So our take homes from this are that metagenomics, metabolomics, and transcriptomics together are shedding light on new targets for treatment of IBS. And in general, this whole pathway that comes together is that increased degradation of purine nucleotides by the host and microbes leads to increased metabolic stress in colonic tissues, and that in turn leads to increased purine salvage in the host cells. And that's it for my case study. So I will head over to Greg and let him present his slides.
Thank you, Tanya. And also, I would like to thank the organizers for the opportunity to present our case study and be part of this great panel discussion with my esteemed colleagues. So, leading the metabolomics for Metabolon, we've really seen the utility of using metabolomics to provide functional insights for microbial activity. And everything Tanya just described kind of represents a quintessential study in how microbial activity is altering gut activity and gut profiles, resulting in gut dysbiosis and some syndromes related to gut disease. But one thing I want to stress is that disease is really systemic. So all of the microbial outputs in terms of metabolism and metabolic outputs that microbes are producing in the gut really have systemic effects, because these can enter the circulation and have wide-ranging effects on all diseases because they can affect all tissue types. So the case study I'm going to be presenting today is an exemplar of that, talking about how gut dysbiosis, and changes in microbial composition can affect distal functions, and in this case, neurological functions by manifestation through ALS or amyotrophic lateral sclerosis, a degenerative neurological disorder. And in this model system, investigators at the Weizmann Institute, Aran Segal and Ron Ellenav, used a model system of superoxide dismutase transgenics that mimics ALS function in this preclinical mouse model. So relative to wild-type mice, the transgenic mice show severe profiles of ALS and ALS-type symptoms. And they suspected a microbial influence in the development and severity of disease. And so they tested this by doing wide-ranging antibiotic knockout of the gut microbe and the microbial environment. And they found that in this model, they had a severe progression of ALS-type symptoms, so that by altering or knocking out microbes, severely progressed the disease in the SOD transgenic mice, but not in wild-type mice. And that's reflected in a neurological score, kind of an assessment, an integrated score of motor activity in these mice in that first center panel there, showing that relative to wild-type mice that are essentially flat along the x-axis, ALS mice in the blue line have a very severe neurological score. The higher the score, the more severe the disease, and that when you actually are able to investigate the basis of what the knockouts do, what the compositional profile is, they actually identified through metagenomic sequencing and shotgun reads the compositional changes in these transgenic mice. So they identified specific strains of bacteria that are either influencing, exacerbating the disease profile, or improving the disease profile when you treat them with antibiotics. And they're able to supplement these mice in monostrain ways, introduce these in single strain versions back into the mice, re-inoculating these mice, and looking for the effect of these mice. And they found several strains that were either exacerbated or improved the profiles. And one of the most dramatic strains that when there was depletion they found through their metagenomic reads was Akkermansia muciniphila. And so depletion of this resulted in exacerbation of symptoms. So they thought that this could have been a beneficial profile or beneficial strain in the treatment or in the development of this disease, and that by supplementing mice in monocultures with the strain, they showed improvement in the strain or improvement of the scores, which is the red line there. And metagenomics are very powerful, and I don't mean to minimize all the work that's done from this. From that first slide where I talked about the study design to that first slide there, because there's an enormous amount of metagenomics information that was useful to identify the compositional changes in transgenic mice and seeing what strains were both exacerbating and improving the disease. But the metagenomics information can only get you so far because it's not really providing functional outputs. And certainly there are programs and tools out there like from the Huttenhauer lab, Human and Metaflin, that can provide some functional insights, but they're mostly in silico and they're limited in their approaches. And where metabolomics is particularly useful is that it's providing the functional readouts of changes in the metagenomic composition of profiles. And because they understood, the investigators understood what changes, what strains were involved in this, they could actually study these mice that were inoculated with these different strains and look at the differences in their metabolic outputs in the circulation of the mice. So changes in gut profiles and readouts in the circulation are what investigators are looking at in terms of the metabolic profiles or metabolomic profiles that we performed at Metabolon. And they found that relative to PBS-treated mice and even wild-type mice, that SOD mice or the ALS mice, the diseased mice that were mono-inoculated with Akkermansia had significantly higher levels of nicotinamide and all the nicotinamide derivatives that are in that pathway. And that's really the power of unbiased or untargeted metabolomic profiling, because you get an unbiased view of metabolic changes across all different biochemical pathways. And here we identified, the investigators identified specific perturbations that Akkermansia muciniphila was able to convey in the nicotinamide pathway, so that when mice were supplemented with this strain, there was a dramatic increase in the nicotinamide molecules. suggesting that these could be playing a beneficial role in the treatment or outcome of the disease. And so they tested this directly. And there's a lot of work that went on between the center panel, that heat map, and the follow-up administration of nicotinamide, because they actually looked at a huge number of strains, specifically seeing in untargeted metabolomics what the differences in the outputs of these strains were in monoculture. And it turns out that Akkermansia muciniphila, as described in the paper, produced nicotinamide at log order magnitudes higher than other strains that were relevant in terms of the changes in the compositional profiles in the microbiome of these mice. So they knew nicotinamide was potentially a high value candidate as a metabolite that's altered and could be supplemented potentially in the treatment of this disorder. And indeed, in the next slide, they treated these transgenic mice with nicotinamide and saw a vast improvement in the neurological score. And of course, this is a preclinical model, so bringing it back to humans, they wanted to test the clinical relevance of this. So they looked at both the serum and CSF that were isolated from ALS-afflicted patients relative to age-matched and gender-matched controls. And that's in the last panel there, showing that ALS patients, in fact, do show evidence of a depletion of nicotinamide. And of course, this is under current investigation to see if this could have high clinical utility in terms of the treatment of ALS. So this single case study provides kind of an example of showing that microbial activity in the gut produces systemic changes, even neurological changes, and that these metabolites are crossing the gut barrier, whether through gut permeability or active transport, entering the circulation, and even crossing the blood-brain barrier to cause changes in neurological disorders. And it really takes a multi-omics approach to understand this, to see the compositional changes that happen in disease, but still it's necessary to understand the functional outputs that come from an untargeted metabolomic platform that can provide insights into the perturbations that are underpinning these changes in compositional profiles. And so really, it's the analysis of these data, so generation of these data, having these multi-omics data at our disposal, but then the next level is how do we bring these multi-omics data together to efficiently analyze them, to understand the metabolic and the molecular basis of disease. And that's why I pass it over to my colleague Damon, who's going to talk about some of those tools and machine learning approaches that are being developed today to help do that very thing. Thank you.
Nima, feel free to share your deck.
Yeah, having some technical difficulties, but it's coming up.
Yeah, we can see your deck now.
All right. Thank you for having me here. Glad to show you one case study, and then we can chat more about the details of it afterwards. So we are a fairly young laboratory at Stanford. We are 4 years old now, and we work on a broad range of translational problems, mostly in the maternal and child health domain, but also other areas as well. We are focused on not just omics data sets, single cell analysis, and various omics integration problems, but also wearable devices, electronic health records, global health level data sets. The case study that I'm going to show you is about pregnancy. Preterm birth, particularly, is the single largest cause of death in children under 5 years old, and it's growing globally. If we could understand what is happening biologically during pregnancy, we can intervene in that process and hopefully improve outcomes of these neonates all the way into adulthood. During pregnancy, the immune system is striking for a balance between pathogen defense for the mother versus immune tolerance for the fetus. So if we could understand what the normal progression of the immune system during pregnancy looks like, hopefully we can use that to detect abnormalities as well. So the first study that we did was a single cell study using a mass cytometer, which is a kind of next generation flow cytometer. It can measure a broad range of cell types. Each dot represents one cell type, two dots that are closer, or two cell types that are correlated across all patients, all time points. But we also can measure various signaling markers in every individual cell. We can have the same signaling markers in response to LPS, interleukin, interferon alpha, to kind of see how the immune system can respond to various challenges. And that forms a complex correlation network that represents the immune system. And we can build a machine learning algorithm that would look at this data and first, in normal pregnant women, predict gestational age at time of sample collection. And then we can validate it in an independent cohort and hopefully use that to detect preterm birth cases as well. So this was the first part of this study that was published a couple of years ago. But what we were really interested in was understanding how the immune system interacts with other biological modalities that we can collect from the same patients. So we put a big consortium together at Stanford where we enrolled the same group of pregnant women, and we measured in the same cohort cell-free RNA in plasma. We had a targeted proteomics assay developed by Somologic. We had metabolomics analysis of plasma also. We had our single cell profiling of whole blood, microbiome analysis from 5 different body sites, and plasma and serum cytokines. And our hope was to put all of these together to build unifying predictive models. But when you have a study like this, the first question that people ask is, well, what correlates with what? And the answer to that is that a lot of various measurements correlate with each other when you measure them in the same set of patients. So you have to take a more principled approach. You can look at ridge regression, where you have your outcome, you have your data, you're trying to minimize your error rate. But it turns out that because the data is extremely high dimensional and you want the models to be translationally relevant, if you change that 2 to a 1, it makes your models sparse, easier to validate, easier to interpret. That's all good. But it turns out that doing this in isolation is not enough. Putting all of these data sets into one of these machine learning models, and not just Lasso, but any machine learning model, ends up not working very well. The reason for that is that there is large discrepancy in various characteristics of these data sets, including size. A transcriptomics data set is really large. In contrast, a targeted proteomics data set is very small. And if you throw all of these into the same machine learning algorithm, you end up completely overwhelming the smaller data sets. So, but accounting for size alone is not enough, even if it would do that. Here is an example of a large data set. Even though it's large, it only has 3 major clusters of features. This is a smaller targeted data set. Even though it's small, there are lots of individual clusters that we can see. There is more information content in the smaller data set. And this is not entirely surprising. We can quantify this. This is size. You have already seen this. This is the number of principal components that I need to account for 90% variance in each data set. And you can immediately see that the smaller ones, even though they're smaller, they have more information content. You need more variables to account for all the variance in them. And this is because these smaller data sets are carefully designed targeted assays that have very little redundancy, as opposed to an untargeted assay that measures the same pathways over and over again at different points. So we came up with a more sophisticated machine learning algorithm that analyzes each data set independently, extracts the relevant characteristics from them, and then a higher level model combines all of those to make the final prediction. And once we did that, things started to make sense. Our final models were more accurate than models that we could build using any of the omics data sets in isolation. And we were able to look at various biological correlations and crosstalks across different omics data sets. For example, it turns out that the STAT5 signaling in T cells is correlated with a module of oral microbiomes of all things, oral microbiome measurements. We then took this Stanford-only model and we expanded it to 5 different low and middle income countries working with the Gates Foundation. Because we had 5 cohorts this time, we would take each data set, build a model specifically for that one cohort, then optimize it using data from all the other ones, tested on the half of the data set that was blinded, and we were able to reach a pretty impressive area under the curve for prediction of preterm birth early during pregnancy. And when we look at the biology of the features that are getting picked up, it generally makes sense. As you move to the right, you have GCSF, PDL1, IL-6, IL-1RA, so generally a module of inflammatory proteins. And yes, you can read about the details of this in JAMA Network Open. Now we are repeating the same thing, in this case in preeclampsia, but we are also doing this in surgery and cancer research and various other areas as well. And the strategy seems to be working pretty well. We can also integrate this with social determinants of health. In this case, we were able to integrate it with stress information in the mother, and that made the models even more accurate than before. And finally, as we do more and more of these multi-omics measurements, the dimensionality of the data set increases, but the number of patients don't necessarily increase due to budget and other real world limitations. So we came up with a pipeline to try to address that. In a traditional bioinformatics pipeline, you have your samples, you measure your features, and your features form a correlation network. You do hypothesis testing, and you hope to get a p-value of smaller than 0.05. That's Carlo Bonferroni reminding you that you need to adjust your p-values. But you can't always just test one hypothesis at a time and increase your cohort size to account for the statistical power that you're losing by adding more omics to the model. So the strategy that we came up with was to mine the literature for prior knowledge about interactions between the features that are being measured. And then we found a way to directly encode that, that prior knowledge into the model itself. You can think of it as a Bayesian prior, and that ended up making our models more accurate than before in multiple data sets in parallel. This was also published a few months ago if you want to go into the details of that. And I think I can stop here. Thank you very much, and we'll talk about your questions during the panel discussion.
Thank you, Ivan. Great presentation. So we receive a couple of very specific questions for some of the projects, and also some other questions that are more general. So maybe we could go first with the specific ones. I have one for Tonya. And this particular one is, has anyone in the field looked into correlation between gout incidence and IBS? And this is because the relationship of a uric acid buildup that is related to purine xanthine pathway.
Yeah, that's a really great question. That's not something I've personally done a deep dive into, but I do know that there is a correlation between gastrointestinal issues and people who are suffering from gout and other kind of rheumatoid arthritis conditions. So it does seem like there could very well be a connection there. And I know there are some drugs that were used to treat gout that were being tested in those with IBS. I think like allopurinol, I think is one. So I think it's definitely something that, you know, we should look into if someone's not already doing that, but I have a feeling there might be some exploring that area right now.
Great. So next question is for you, Greg, and this relates to Akkermansia. Have you tried to isolate or quantify Akkermansia municipalia that are already present in ALS mice to avoid any bias?
That are already present. Oh yeah, so the compositional profiles are different in ALS mice already. So the investigators did a lot of work. So the LNAV lab did a lot of work showing vivarium-dependent changes in compositional profiles and how that relates to disease severity. So all that was checked, and there are differences in the mice. So the disease does seem to promote a different compositional profile at baseline, that there is less abundance of strains related to Akkermansia muciniphila and nicotinamide producing strains, but Akkermansia was the one that was the most potent in this regard. And there are compositional differences that result in the depletion of this strain relative to control mice at baseline. Yeah, it's a great question.
Perfect. If anybody has a specific question, just keep them going. So we have some that are more general, and I think that will drive some of the discussion. And this is one, I'm interested in incorporating multi-omics approach in my research, but I cannot afford to pay for all the omics that I can imagine. how can I choose between the different noms? What would be the first one? Thoughts?
Well, that's really kind of a related question that I had for Nima too, resulting in the predictive accuracy of the models. Do you have a sense for which omic streams may be most potent or contributing to the accuracy of those models? So that kind of feeds into that question, I think.
Yeah, of course. It really depends on the outcome that you're looking at. When I only showed you preterm birth results, but and in those cases, inflammation is important. And the proteomics assay is doing really well. But if you try to look at something like preeclampsia that has a cardiovascular component to it, the metabolomics assays become a lot more important. So the short answer is that I would try to use a broad range of omics assays in a small number of patients and use what you learn from that to inform the design of your larger study. There is really no easy way to come up with something without having any prior data. That being said, the more modern targeted proteomics approaches are able to measure multiple panels that capture different aspects of health. If you had to go with just one thing, perhaps that's the place.
Okay, that's great, and we will talk about more in terms of study design and sizing a little bit after some of these questions. So there's another one here that I'm working on a systemic disease. How I know if a microbiome or microbial derived compounds are relevant to me. I think that relates to the first one. So I think that the question is, some of these, for example, Tonya, you talk about gut microbiome, that is a representation of disease. What's the relevance of that in a systemic disease? So should we explore gut microbiome if I'm interested in Alzheimer's, for example?
Yeah, I think that's a really good question. And also, one that is definitely dependent on the context of what you're studying. So if there is prior knowledge that you think there could be a link between something, say like Alzheimer's and the gut microbiome, then definitely worth exploring further. But also if we think about it in terms of the role that the microbiome might be playing directly or indirectly on things, sometimes it can also be worth exploring just on that avenue alone. So in terms of processing drugs that might be used to treat different diseases like Alzheimer's, we potentially would want to know if your microbiome might be changing things there as well. So I definitely think it would be warranted if there's prior knowledge or some sort of hypothesis linking the two together.
What about the microbial derived compounds, Greg?
Yeah, what I would add to that is kind of an extension of what Tanya was saying and what we were discussing earlier, is that gut microbial activity has been shown to affect every organ. So there's just a huge number of papers now supporting that gut-derived microbial activity is influencing every organ and therefore every organ disease, pretty much every disease that can be out there. And these are systemic diseases. So gut microbial activity is obviously influencing these organs either through receptors in the gut and also through entering the circulation, either through gut permeability, dysfunction and barrier dysfunction, or active transport from receptors and transporters. So that it has to be a necessary component, I think, of everything that is being looked at, because there is potentially a huge role in this. All these papers that have come out recently and information showing that it's essentially the new organ, that you have 2 pounds of bacterial mass that are in your gut that are influencing every disease process and the metabolism of drugs, and it's influencing response and non-response and outcomes. So it, I think, has to be a central component of everything that is currently being looked at. And fortunately, we have the tools now that we're able to collect these samples in a standardized kind of way and bring them into an analysis platform to look at these and all these omic streams in a very careful way. And I think it needs to be a central part of every research plan now.
That's great. So if we think about the study design and integrating and samples, so let's focus a little bit on that aspect. So Nima, a question for you is, how do you calculate that sample size to power your multi-omics study in a proper way?
Yeah, there is no easy way to do that without having preliminary data. You have to start from somewhere, collect some amount of data, see what your effect size is going to be. And based on that, you can do a careful power analysis to determine the size of your eventual cohort. But without having preliminary data, there is no way to have an effect size unless you're willing to look at the literature and make assumptions about what your expected effect size is going to be, but that's a pretty risky. So just like the answer to the previous question, my advice is to just get started with a pilot analysis, and based on that, determine the cohort size.
Yeah, and I think that relates to the next question in terms of how could I design my experiment, my study? So how I define my groups, so how I define my inclusion criteria, my exclusion criteria, and so on. So I think that that's a question for all of you guys that have done this type of experiments. So maybe we could start with Tonya. Nima, it's okay. Yeah, you can go for it.
Just a brief note. All right, thank you. I'm sorry for interrupting. My brief thought on that is that it has to be driven by the clinical process, by the question that you're actually trying to answer. Where would the impact for that come from? What is the patient population that you're trying to help? And then if you had a successful multi-omics analysis, how would it inform clinical decisions? Those are the questions that I would think of.
Sorry, Nima, we missed the last part. Could you repeat the last two sentences?
Yeah, I would just think about how a successful multi-omics study would guide clinical decision making or the design of new therapeutics. Based on that, I would select my enrollment criteria.
Tonya, Greg?
Yeah, I would totally agree with that. So generally speaking, when we're designing a study or helping someone design a study, we always try and work our way backwards. So based on what we are anticipating, like some sort of measurements or outcome to be, how can we best assess that using a certain population or certain study design, whether it's longitudinal sampling, cross-sectional sampling, and which ways can we measure, and what will those outputs be, and how can we determine, their interactions. That's going to play a role in how we end up enrolling people, which types of samples we collect, and how we preserve them, and how we measure different metrics from them. So we always try and work our way backwards, if that makes sense.
Mm-hmm. Yeah, I wouldn't really have anything to add to that. That's exactly right, because you start with your effect size, where you want to be, what you're trying to analyze, what your outcomes are, and based on how strong those effects are, that dictates what the power should be for that. And there's, I think there's a whole army of biostatisticians now, bioinformaticists, who can provide power calculations based on these that can answer these questions. But it is central to everything you want to do. You want to have an appropriately powered study to ask the question. You don't want to necessarily need to enroll 1,000 people if you're asking a pretty simple question with large effect sizes. So economy and the research focus needs to be regarded here. But I think I agree with everything my colleagues just said.
Yeah, and I think that was a good segue to a couple of questions that we got, and in terms of the sample and what to analyze with them. So one question is that how can I be sure that the information that I've generated resemble what you could find in the individual, in the donor? So how do you preserve that in vivo profile from the beginning all the way to the end? So Tonya, maybe.
Yeah, so making sure that you don't have like any biases introduced in anything that you're actually measuring along the way is definitely a great concern. So I would say one way to make sure that that's happening in the least amount possible would be to really make sure that you're using either collection methods or storage methods that are really appropriate for the samples that you're using. So if you're collecting biopsies, for example, how you store them and how you process them can be completely different than how you would treat something like a stool sample. So again, thinking kind of backwards in terms of where you're going to be using this data and how you're going to be using it is going to impact how you collect it, store it, and make sure you're not introducing bias up front that you're going to potentially interpret as a signal on the back end. So there's definitely many different ways to go about that. One of the things that I've found to be the easiest in terms of sample collection, at least, is when we can collect and measure in one kind of way. So like if we have metabolomics and metagenomics, potentially metatranscriptomics, if those are all things that we think are going to be impacted, being able to get those measurements from just one sample and making sure that that sample type is not introducing any sort of bias to one of those measurements is one of the best ways to go about that.
Mm-hmm. Yeah, I think that relates to a specific question for Greg. They are asking, like, could you look at gut activity using urine as a sample type? Is that something that you do or you would suggest, Greg?
Yeah, so a few things here, and of course I welcome comments from my colleagues as well. Metabolomics is unique from genetics, for example, because your genes are your genes, and regardless of the cell type, your DNA is your hereditary material, and it largely is unaltered across all sample types. And that's not true for metabolomics and metabolism of different samples, so that stool sample is going to be have very different metabolic profile than a urine sample, than a serum sample, than a CSF sample. So it's, for metabolomics particularly, it is imperative to get the right sample type to address the appropriate question at hand. To this specific question, we have done a great deal of work with urinary metabolomics, and urine is actually a very rich matrix metabolomically. And it is a wonderful surrogate for gut activity and microbial activity. A lot of those are excreted through a phased elimination in the body, and they are represented in the urine. That said, it's a little more difficult because it's not a homeostatically maintained matrix or sample type. So in terms of comparing directly across different samples, some very careful controls need to be taken into account, and we have a lot of guidance in terms of providing that information so that samples can be compared within a study and then even longitudinally. But you need to be paying very special attention to that. And if that's what's available, that's what's available. But to look at gut activity, obviously the gold standard, I think, would be stool, and to have the ability to collect those samples in a very standardized way for analysis that doesn't introduce confounding, like Tanya's points, is important. But yes, so that it can be done.
Okay, that's great. So now, if we start thinking about, we pass the logistic point, so we have the proper sample, we have we start generating data. So now we have the data, and that raises a lot of questions. So I have one here. What are the major challenges for multi-omics data integration? So maybe we could start with Nima.
Yep, sure. So one of the biggest challenges is missing data. Because if each of your assays fail in a limited number of patients, that's fine when you're analyzing that assay. But when you try to combine different omics data sets, the missing information compounds across all of them. So you have the patients that end up having all of the assays become very, very rare and very valuable. So that's one place that I would look at. One of the biggest limitations is what do we do with the amount of missing information, and how do we control for it? The other challenge is the one that I talked about, differences in sizes and modularities and correlation structures of these data sets, because we have very good pipelines for microbiome analysis and for metabolomics analysis. These have been optimized over many years, but we still don't have good pipelines that would work consistently for all of these omics or for the combination of them. So that justifies the need for that two-stage approach that I presented, that you would first extract features from each of the omics data sets, and then you would try to combine them. And the last part is the complexity of accounting for crosstalk between different data sets. It's one thing to just throw all of them into a machine learning algorithm and make predictions of outcomes. It's another thing to try to find crosstalks that represent mechanistic biological connections that can be used as a target for an intervention, for example. And those correlation networks end up being really big and difficult to calculate. We need new methods for doing that more effectively.
Tonya?
Yeah, I would say one of the biggest challenges is, and Nima definitely commented on this already, is really understanding the structure of your data. So in order to find signals that are real, you have to have a really great understanding of how to use the types of data that you have. So like if you have compositional data, treating it accordingly, and making sure that you're not just kind of ignoring or washing over some of the principles that you should be applying when you're working with that data. And it is a little bit like the Wild West right now. You know, there's like new things coming out all the time in terms of analyzing data and using it effectively, but there are definitely some statistical principles that should never be overlooked. And so finding an expert like Nima or someone in biostats that can really help you understand how to use the data correctly so that when you do find signals, they're actually meaningful. I think that's like the #1 for data integration from my end.
Greg?
Yeah, the only thing I would add to that, and that's been really well covered by Nima and Tanya, is that depending on the data stream available, what omics are available, that there could be potential confounding from crosstalk between the omic streams, so that proteins obviously affect the levels of metabolites, and metabolites can affect the levels of proteins. That's why genetics is such a powerful tool, because typically the downstream omics modalities don't affect the upstream genetics. So your genetics is an unchanging instrumental variable in that regard. But in the absence of that, there could be confounding from the crosstalk between these omics affecting each other, and that needs to be paid attention to. I know there's some tools out there, and there's thought given to that approach and limiting that confounding, but that's something else to be paid attention to, for which should be incorporated into a research plan.
Okay, that's great. So another question related to this is, how do you validate your multi-omic data sets to ensure they are accurate, reflect the biology and the physiology of what you are studying, and how you establish those correlations? So how you validate one of these data sets?
Well, I would say, please, Nima.
No, go ahead, Gary.
I was going to say a validation. I mean, part of what we do is you know, and Nima can talk about this or acts eloquently, I'm sure, from a bioinformatics perspective and machine learning perspective, to have discovery and training data sets, and you can divide things, and there are supervised approaches from the informatics and computational side to do this. From the biological side, which is my background, it is discovery and validation sets and cohorts that you'd be working with, so that they would be tested in distinct cohorts, whether preclinical or clinical in humans, to look at discovery and validations to make sure that you're following valid biological signals.
I agree with that. I think having an independent validation cohort is very important, but I also understand the cost and timeline of that is not always very convenient. So what you can do in the meantime, while you wait for independent validation, is comparison with prior knowledge. If there are positive or negative signals that you expected from before, and you end up seeing those signals, that also ends up being quite reassuring, not a replacement for validation. The other idea that I would think about is to include positive and negative controls in your data set from the beginning, from when you were designing the experiment. And then those positive and negative controls can tell you a little bit, at least, about your limit of detection and about whether everything is working the way that you would expect.
And related a little bit with what we are talking about in terms of validation of pipelines, but that extends to all the steps. And the question here is quite interesting. It says there is a lot of information in public databases. However, I'm not sure how the data was obtained. So how can I trust the public domain? Pretty much that's the question. Tonya?
Yeah, that's a really good question. And I think I think there are steps that are moving us as a whole towards, a better like public repository scenario. So making sure that your data is fair, right? So the fairness principles of having it accessible, reproducible, having the appropriate metadata in the right formats associated with everything submitted. If something is not following the FAIR principles that are put out by the NIH, then it's not data personally that I would feel necessarily comfortable using. So I think for me, it comes down to making sure that the information I have regarding a data set is as accurate to my knowledge as possible, which usually comes through the most transparent mechanisms of having all of the metadata there with it at the time of submission to something like SRA or ENA.
Mm-hmm. Great. Thoughts?
Yeah, and I would add to that, as I have an academic background, I ran a liver pathophysiology lab for many years, and we mined publicly available data for a number of years trying to identify concordant signals in our own data, and we had trouble. So the question is very well taken, and it is an issue, I think. And to Tanya's point, so long as you are going after the best data from procured data that follows all the proper adherence guidelines is foremost important. And also to be able to identify the data sets that most closely resemble your model system. So there are obvious and honest differences in data, and that's why we don't have a concordance of understanding across all the diseases, because there are a number of differences in all the reported literature that make it unclear of the true mechanistic basis of illness and many diseases. And that's because there are honest differences in model systems and how data were collected and how they were analyzed, so that we really need to pay attention to what controls were used, how they were used, what are the confounding variables that need to be accounted for. Maybe they weren't accounted for in a given study, and that's why there are differences. So to try and find as close a comparator as we can in terms of public literature to find the positive and negative controls like Nima was talking about, to be able to identify those signals that you know should be there. Do you identify concordant signals? And then you have a lot of confidence in being able to extend those with your own data.
Nima, anything else with this respect?
No, I think that's covered.
So now, and I think that relates to this. So the question is, thinking beyond association and correlation, is multi-omics the answer to define causation in disease? I think that's a quite complex question. So in other words, if by adding more omics, are we understanding physiology to the point that we know what's causing the disease or we can prevent it? Any taker?
I can start in just regards to the case study that I presented. So if we were to only look at one ohm, for example, in that study, we would not have a picture around that pathway that I presented. There were too many signals and you wouldn't have been able to find it. Does that pathway mean it's the only cause of IBS? Absolutely not. That's not something I'm throwing out there. But does it give us a target for treatment? Absolutely. So I don't necessarily think that a multi-omics approach is going to be the end-all be-all in terms of having to do away with testing things in model organisms or moving things to an actual experimental setup in the lab to validate certain things. Nor will it ever replace like clinical trials or things like that. But it will definitely help us get a better understanding quicker of the systems that we're interested in my opinion.
Yeah, and I think my case study shows an example of what Tanya was just talking about. Metagenomics were instrumental in identifying compositional changes in ALS mice. That alone was insufficient to understand the basis of disease or what was contributing to severity of symptoms. Nicotinamide was found through metabolomic analysis, but there were a lot of other signals that were identified that differentiated those mice too. So given all of the variability in those signals, to have one emerge to create a high value candidate or hypothesis develop really wouldn't have been possible. You really need both those together to come together to point you in the right direction, at least to reduce the signals to one or two manageable, tractable, testable hypotheses. And to extend that a little bit, I think there are approaches now that have developed recently, or somewhat recently, in terms of using genetics as the key anchor to help identify causal pathways. And that's approaches like Mendelian randomization that you can, by understanding the genetics and outcomes and having typically an orthogonal data stream, something like metabolomics, that can be deployed to help show that genetics alone, given the complexity of disease, is insufficient to show causality in a given pathway. You're only looking at risks of the disease with genetics, right? So you really need an orthogonal data stream to point to show that there's an intermediary phenotype or analyte that can be detected to show also correlation that one is affecting X and X is affecting Y to show causality in the pathway. And you begin to, those causal inferences begin to emerge, and it's because of a multiomics approach that you can establish that. So yeah, it's absolutely needed, and we're beginning to get there, showing causality.
Any other thought, Nima, in terms of causality?
Yeah, sure. I think you can use a multi-omics longitudinal study to leverage that element of time, particularly to think about causality, not in the classic statistical sense of it, but in terms of what events happen before which and when there is a one-way correlation, which people sometimes refer to as causality. But ultimately, once you have those candidates for causal link, nothing replaces a carefully designed experiment that tests whether A causes B in the most stringent. So you can come up with hypotheses about causal connections, but you do have to validate them independently.
Perfect. That's great. So, any final thought for people that are trying to do multi-omics? So, if there's something approach to you, what's the first thing that you tell them to do? Yes, I'm interested in doing multi-omics. What would be the next step? Jonia?
Well, I would definitely ask them what their hypothesis is. That's always my first question. And then from there, we can work our way backwards to figure out what we would need to get you there to be able to try and answer your research question in particular. I know that's kind of a blunt answer, but.
Greg, anything in particular that people need to know before even considering multi-omics?
Yeah, what is your biological question? I mean, that is central to everything, because that'll tell you what samples need to be involved, what collection protocols need to be involved, how the data should be collected, and how they should be analyzed. So that is the first question that needs to be addressed, and a very clear study design and hypothesis.
That's great. So I think that let me see if we have any other questions coming there. In the meantime, there is one here for you, Greg, quite specific. It says, what are some methods you can find useful for on-target metabolomics in regards to annotation to peaks of the data and bypassing annotation to get the insight results? That's the question. I don't know if you got it.
That's I'm a little confused by that. Part of what makes metabolomics so useful is that we have the annotation of the metabolites, right? So unlike genetics and even proteins, genes and proteins, where we still have massive gaps in knowledge regarding the function of these genes, the function of these proteins, they have names, but we have no idea what these things are doing in large regard. Excuse me. With metabolomics, the power of metabolomics is that we rest on 200 years of metabolism research. So we know what these metabolites are, we know their structure, we know these are largely conserved down to bacteria, and that structural conservation implies functional conservation. So these are very high degree of translatability across model systems, across the phylogenetic spectrum because of that, because we understand the annotation. So we can't take the annotation out of it. That's central to everything that makes metabolism useful as an analytical tool and an omic stream. Regarding the peak assignment, we know this because it comes from an LC-MS technology, at least that we utilize. And we believe this is the most powerful approach for unbiased metabolomics. And this is predicated on kind of the largest chemical reference library that we know these peaks because we are able to compare these to standards. So we know exactly what they are, and we know what their structures are, and then we know what their structures are, we know what their function is through the annotation. So there's no easy tool out there. And I know there are some peak alignment and peak assignment tools out there, but from my experience, they are weak. And part of what makes what we do so powerful and our technology platform so powerful is that it's based on these structural assignments based on chemical reference standards. So there is no easy answer, I guess, to answer that.
Perfect. I don't see any more questions. If anybody that is listening is still interested in getting a question answered, just send it my way. Otherwise, I think that this will conclude the Progress on Omics in Microbiome Research webinar. We thank the presenters. I think that the use cases that you share with us today truly demonstrate the value of doing multi-omic analysis, but not only doing multi-omics, it's thinking from the very beginning, design the studies in a way that will try to answer your biological question, doing pilot studies just to try to see which of the omics are more relevant for your research, and after that, find the right type of experts to help you designing the study, trying to calculate the study size, and so on. It says here, can Nima say more about size? I will go back to that one before we go, but clearly to my point, so talking to the experts is key here. So final question, can Nima say more about size versus modality and how having less data can improve prediction versus having more data?
Yeah, of course. So the way I like to think about it is in terms of biological pathways. If you have 20 major cellular pathways that you need to look at, you have the option of designing a careful panel that looks at those 20 pathways in a validated way using assays that have been reproduced before. Or you have the option to use an untargeted RNA-seq that would measure everything, including those pathways redundantly at various points. What our studies are pointing to is that fewer measurements that are carefully validated, or at least in the studies that we have done, are more valuable than measuring lots of various features and hoping that the machine learning algorithm can figure it out later on. This also becomes important in the context of some of the other topics that we discussed. How do you call the peaks of a untargeted metabolomics assay? You have the option to validate every one of your peaks one at a time and make sure you really understand what they are. But you also have the option to take the entire raw mass spectrometry data, and there are machine learning algorithms that will just process that automatically. Especially in these smaller data sets, when you have tens or hundreds of patients and not thousands of patients, making sure that the quality of the measurements is really high becomes important. Once you go into thousands of patients, you can hope that the machine learning algorithm will be able to figure it out independently. But my expectation at this point is that in a smaller data sets, you want higher quality data. That's to the extent that's possible.
Okay, that's perfect. One more for Greg here. How would you study the gut microbiome? You know, which type of metabolomic study would you design for gut microbiome? It's a tricky one.
Well, it goes back to what Tanya was saying earlier. What's the hypothesis? What are you studying? Do you know an involvement, a specific question that you're asking? For example, if you're studying something more systemic, you'd want to capture samples of what that disease or that tissue reflects. So A, start with your hypothesis and what your model system is. But B, also that will inform what your sample collection should be, so that if things are focused in the gut and it's a gut-derived disease, gut dysbiosis, IBD, IBS, Crohn's, then I would think you'd want GI-focused collections, and you'd want those collected in a very standardized way. And part of what makes studying gut metabolism so powerful now is that we have the approaches, the sample collection protocols to be able to collect these samples and address these biological questions in a real way, because we're going to have validated, standardized samples that can be taken across any analysis platform, whether you're looking at metagenomics, metatranscriptomics, or metabolomics, that we have the protocols in place to be able to collect these samples and analyze them and then compare directly across samples and even longitudinally. So study design and informing sample collection is what I think are paramount to a successful study. And of course, any of the panelists, my colleagues, would be happy to discuss more deeply within the different platforms in which we work.
Yeah, I would just add on to that, like if you're specifically interested in the gut microbiome and the metabolites it might be producing, definitely taking like a two-step approach, like maybe potentially looking at changes in metabolites in general first, and then using metatranscriptomics or metagenomics to see is that correlating with specific fungal taxa or their genes could be an approach to dive a little deeper. Something we're trying.
Perfect. Now, a very timely question here is: has anybody looked at an association between the microbiome and COVID-19 in healed and dead people? I'm just reading the question. Tonya?
Yeah, okay, well, that's an interesting one. So I know that there have been some studies looking at the gut microbiome in relation to COVID-19 infection. Personally, I haven't been involved in any of those studies, so I'll throw that out to Greg or Nima in case you have.
Yeah, so I know it's being looked at. I don't know the results of those, but look, just as we talked about, it's going to affect every disease profile or the arc of every disease. So of course it's being looked at, and I know there's a national study looking at that right now called the IMPACT study, looking at changes across all organ systems, inflammation, microbiome, microbial activity, et cetera. But again, I don't know of any specific results that speak to influences on outcomes.
I think that was the last question that I have here. So again, I really want to thank the presenter for this exciting webinar. I think that we all learn all aspects of multi-omic design. So talk to the expert. Clearly, they will help you to answer your questions. And for everybody that listened to the webinar, we also thank you for all your questions. And hopefully you are, as me, very happy to be informed of multi-omics and how we could use this approach to understand biology and hopefully find better answers and treatments and biomarkers in the future. Thank you very much and have a great day wherever you are.
Great. Thanks, Mark. Thanks, Carlos.
Thank you very much, Carlos. Just jumping on quickly, just for a few housekeeping notes for the audience. But Carlos, thanks so much for facilitating this. In terms of what's happening next, all the audience will get an email just to, it should be within the next 24 hours with a link to this on demand. So if you want to watch this back, that's possible for a period of time. It will include in there, I think there's a link to feedback. So if you anything on the webinar experience, you can let us know. And also we should include some links to other resources and downloadable content. So please keep an eye out for that. That aside, yeah, just like to reiterate once again, Tonya, Greg, Nima, thanks so much for jumping on and sharing your work. And obviously DNA Genetech and Diversigen, we really appreciate your support in this, because obviously it couldn't happen if we didn't have some support to put this together. And obviously collaborative working is so important. So working with two sponsors side by side, you know, it being collaborative in conception really helps us. So thank you very much. And I'll end the session now, but thanks everyone for joining us today.
Great, thank you.
Great. Thank you.
Right.